Echolocation, the use of echoes and sound waves used to determine where an object or objects are in a space. It is based in the neuroanatomy of auditory brain circuitry. Brain pathways allow to calculate the difference of sounds between ears down to fractions of a second. Echolocation is used in different ways by many animal species. It is generally done by ranging the delay of time between when a sound is emitted and then any echoes returned within the environment. Echolocation of bats and dolphins is an extraordinary case of convergent evolution. Genetic studies in this area have found that the evolution of nucleotide sequences is associated with voltage metallicity during the cochlear amplification of signals. This involves complex mechanisms including signal recoding, nerve transmission, and cochlear amplification. Bats use echolocating to assist in both feeding and flight. Echolocation is the process by which sonar is used, signals are emitted and returned to allow the bat to maneuver in little or no light conditions. The sonar allows the bat to see in azimuth, distance, and elevation to where a food source is from their location. Azimuth is the direction of an object from the observer, which is expressed as the angular distance from north or south with respect to the horizon. Bats have the ability to perceive a change in angular direction to as little as 3 degrees arc vertically. This sensitivity in change of angular direction is influenced directly by the deflection of the tragus within the bat's ear. Due to different species of bats having different ear designs, this sensitivity varies between species, where some species have a greater acute sensitivity compared to others. This detection technique has been acquired by bats due to echolocation being far more effective means of detecting structures and food sources in little or no light conditions compared to that of relying purely on eyesight. Echolocation calls are mostly ultrasonic, where the call's frequency is between 20 and 200 kHz, which is produced by the bat's larynx. The sounds are emitted by a group of receptor cells which belong in the inner ear. This allows the bat to detect the slightest change in frequency, thus alters the bat's vertical positioning. Ranging is done by measuring the time delay between the animal's own sound emission and any echoes that return from the environment. The relative intensity of the sound received at each ear, as well as the time delay between arrival at the two ears, provides information about the horizontal angle, azimuth, from which the reflected sound waves arrive. Dolphins and other marine mammals also use echolocation. They do this by generating high-frequency vocals through their nasal passages. This is a constricted area underneath the blowhole. This mechanism is more complex than vocalization of other mammals and uses not the larynx like bats, but air pockets, fat bodies, and large muscles compact into a small area within the face. The dolphin ear is adapted to the marine environment. There is no difference between the inner and outer ear environments. Sound is passed to the inner ear through a fat-filled, low-impedance cavity. The ear is isolated by air-filled sinus pockets from the skull. This allows for superior underwater directional hearing. The dolphin's vocals are high-frequency clicks which come from an organ called the melon, which consists of fat making a large depression. Varying between dolphin species, there are 4 main pairs of air sacs. The dolphin exhales air pressure through these nasal passages, through the monkey lips. The sound then echoes off the air sacs and through the skull, through the melon and then into the water as a clicking sound. These clicks are projected, creating sound waves that bounce off objects such as fish and return via echoes. This allows the dolphin to determine not only the location of the object, but also the size. Each animal, bats and dolphins, live in very different environments. However, each uses echolocation for similar reasons: to find food, judge how far away and how big that food source is, as well as communicating with each other. Bats, however, also use this to find their way around. In terms of range for ultrasonar communication, bats use echolocation over short ranges, 3 to 4 meters, while dolphins use echolocation over longer ranges, up to over 100 meters. The return duration between bats and dolphins is different due to their vastly different environments. The speed of sound is 1/5 of the speed of sound in water, allowing for dolphins to communicate over longer distances more effectively. It is also noted that dolphins do have a larger head and ears in comparison to that of the bats. meaning a bigger distance between each ear. This provides the dolphin with an advantage in using echolocation to locate specific items, as the sound direction can be much more distinct. Both dolphins and bats' genomes have been closely studied to assess any similarities and differences. The protein prestin is found in both bats and dolphins. It is a protein found in the hairs of the inner ear, allowing to rapidly contract and expand in order to transmit signals, which in turn allows the cochlea to amplify sounds. This allows echolocating animals to use sound more effectively over a greater range of frequencies, which is an advantage to both dolphins and bats. In any case, the use of echolocation is an extraordinary case of convergent evolution, which we are yet to find out much more.